Meiosis. Once in the gonad, the primordial germ cells continue to divide mitotically, producing millions of potential gametes precursors. The PGCS of both male and female gonads are then faced with the necessity of reducing their chromosomes from the diploid to the haploid condition. In the haploid condition, each chromosome is represented by only one copy, whereas diploid cells have two copies of each chromosome. To accomplish this reduction, the germ cells undergo meiosis. After the germ cells last division, a period of DNA synthesis occurs so that the initiating meiosis doubles the amount of DNA in its nucleus. In this state, each chromosome consists of two sister chromatids attached at a common kinetochore centromere. In other words, the diploid nucleus contains four copies of each chromosome, but each chromosome is seen as two chromatids bound together. Meiosis shown in Figure 2.9 entails two cells division. In the first division, homologous chromosomes and the chromosomes 3 pair in the diploid cell come together and are then separated into different cells. Hence, the first meiotic division separates homologous chromosomes into two daughter cells such that each cell has only one copy of each chromosome, but each of the chromosomes has already replicated. Each has two chromatids. The second meiotic division then separates the two sister chromatids from each other. Consequently, each of the four cells produced by meiosis has a single haploid copy of each chromosome. The first meiotic division being wings with a long prophase, which is subdivided into 5 stages during the leptotene Greek thin thread stage. The chromatin of the chromatins is stretched out very thinly, and it is not possible to identify individual chromosomes. DNA replication has already occurred, however, and each chromosome consists of two parallel chromatids at the zygote in Greek yoked thread stage. Homologous chromosomes pair side by side. This pairing is called synapsis, and it is characteristic of meiosis. Such pairing does not occur during meiotic division, although the mechanism whereby each chromosome recognized its homologous is not known. Synapsis seems to require the presence of the nuclear membrane and the formation of a proteinaceous ribbon called the synaptonemal complex. This complex is the formation of a ladder-like structure with a central element and two lateral bars. Von Wisten 1984, Schmeichel and Donenholt 1995. The chromatin figures 1913 examinations of meiotic cells nuclei with the electronic microscope. Moses 1968, Moens 1969 suggests that paired chromosomes are bound to the nuclear membrane, and Cumming 1968 had suggested that the nuclear membrane help bring together the homologous chromosomes. The configuration formed by the four chromatids and the synaptonemal complex is referred to as a tetrad or bivalent. During the next stage, meiotic prophase, the chromatids thicken and shorten. This stage has there been called the pectin, Greek thick thread stage. Individual chromatids can now be distinguished under the light microscope, and crossing over may occur. Crossing over presents exchange of the genetic material whereby genes from the chromatid are exchanged with homologous gene from another chromatid. Crossing over may be continue into the next stage, the diplotin, Greek double threads stage. Here the synaptonemal complex breakdown and the two homologous chromosomes start to separate. Usually, however, they remain attached at various places called chiasmata, which are thought to represent regions where crossing over is occurring. Figure 19.14. The diplotin stage is characterized by a high level of gene transcription in some species. The chromosomes of both male and female germ cells take on the lemphorus appearance characteristic of chromosomes that are actively making RNA below during the next stage. Dikinesis, Greek moving apart. The centromeres move away from each other, and the chromosomes remain joined only the tip of the chromatid. This is the stage of meiotic prophase, in with the breakdown of the nuclear membrane and the migration of chromosomes to the metaphase plate. During the metaphase, first homologous chromosomes are separated from each other in an independent fashion. This stage leads to telophase first, during which two daughter cells are formed. Each cells contain one partner of the homologous chromosomes pair. After a brief interkinesis, the second division of meiosis takes place. During this division, the centromere of each chromosome divides during the anaphase, so that each of the new cells gets one of the two chromatids. The final result being the creation of four haploid cells. Note, note the meiosis has also resorted the chromosomes into new groupings. First, each of the four haploid cells has a different assortment of chromosomes. In humans, in which there are 23 different chromosomes, where there can be 223 nearly 10 million different type of haploid cells formed from the genomes of a single person. In addition, the crossing over that occurs during the pectin and diplotin stage of prophase first further increase genetic diversity and makes the number of different gametes in incalculable. The event of meiosis appear to be coordinated through cytoplasmic connections between the dividing cells, whereas the daughter cells formed by meiosis routinely separate from each other. The products of the meiosis cells division remain coupled to each other cytoplasmic bridge. These bridges are seen during the formation of sperm and egg throughout the animal kingdom, bleeding and spreading. 1998.